Welcome to this episode of Life Science Success. For those of you who don't know me, my name is Don, and I'm both a digital marketer and a consultant in life sciences. And before we get started, I wanted to just introduce our sponsor for the podcast.
Episode of Life Science Success is brought to you by D3 Digital Media Marketing. Dive into the digital world with confidence and creativity on your side. D3 Digital Media Marketing is your all-in-one solution for making a lasting impact online. From the vibrant, engaging world of social media marketing, podcasts, and webinars, to the strategic depths of website development, SEO, pay-per-click, email, and content marketing, we tailor our approach to fit your unique brand story. Let's elevate your presence, connect with your audience, and drive success together. Your digital journey starts here. Visit d3digitalmedia.com to learn more.
And so today, my guest on the Life Science Success podcast is Dr. Mohit Jain. Dr. Jain is a physician scientist with more than 20 years of experience in physiology, biomedicine, engineering, computational biology, and mass spectrometry. And I'm really excited to get to welcome you to the podcast. We don't have that many people that do a lot of things inside of this space, and it was interesting as I went through your bio. I have a feeling that we're going to get to dive into a lot of really interesting things that we don't typically cover on the podcast. So welcome.
Thank you so much, Don. Super excited to be here today and appreciate your time.
Yeah, so why don't we just start with, kind of your background, a little bit about you more specifically, and, what led you to Sapient Bio?
Sure. You're going to find this is the classic circuitous case of career development, one in which the individual is searching for what they want to do when they grow up more so than anything else. And so started academically as an MD, PhD, did full clinical training in adult medicine and cardiology. I absolutely loved taking care of patients, loved every patient interaction and the privilege of helping people. My PhD work is in molecular physiology. And one of the challenges that we were struck often with as we were taking care of patients, particularly in the Boston area, you get these very bright people who get admitted to the hospital. These MIT professor types. And, oftentimes they'd ask, why did I have this heart attack? Explain this to me. I'm a 45-year-old guy, exercise, I don't have diabetes, I don't smoke. Explain to me why I have heart disease and why I ended up here in the ICU. And you go through this whole explanation that's based upon what the textbooks say. And they often would time stop for a minute and they would say, you have absolutely no idea why this happened, do you? And that's the legitimate truth. We can give you statistical information across a population, what are the risk factors, but why any singular individual develops disease or remains healthy or what's the best treatment for an individual is not one for which we have great understanding. And this led me to my post-clinical training as a postdoctoral fellow at the Broad Institute at Harvard and MIT, where I spent quite a bit of time in a technology called mass spectrometry. And the real genesis for this stems from the Human Genome Project that had just been completed in the early 2000s as I was finishing up my clinical training. And the human genome was going to uncover everything we wanted to know about biology. Why Don is going to be healthy until 300 years of age and why I may not be, or what's the best drug for me, or it'd be omniscient. It would be the crystal ball that tells us everything about human health. And I remember at that time, we were just, we had the first parallel sequencers. We were sequencing the heck out of everyone and every sample we could understand and get a hand on. And this was really going to uncover the genetic landscape of disease. And once again, this was supposed to be the great promise in medicine. And I remember hearing this incredible lecture in which there was a thought exercise. And essentially it went as this. If we were to sequence every single person on the planet and we had everyone's medical records, how much of human disease could we explain? And this is essentially known as the heritability index. How much of human disease is actually inheritable? How much do we get from your mother and father? What portion of that is encoded in your genome? And what portion is not encoded in your genome? And the underlying belief is that if we sequence the entire planet, we would explain 80, 90, 95% of disease. And the true numbers are probably on the order of 14 to 15%. We know after 1 million genomes or so have been sequenced publicly, that answer is probably in the 6 to 7% range. And so I became very interested in that 90% of disease risks that's not captured in your genome. everything that comes from what you eat, smell, smoke, drink, the way in which you live your life. And we know that that's a critical factor. The standard quote is it's your zip code, not your genetic code, that controls human health. And I was very interested in uncovering that information and being able to probe it in a much more systematic manner beyond just questionnaires. And this what gave rise to my interest in mass spectrometry at the time, in that this was a burgeoning technology that finally allowed us to capture the thousands of molecules in human blood that provide some information about the world in which we live and how our internal organ systems communicate with one another, and perhaps shed light on that 80-90% of missing population attributable risk. And so spent years in mass spectrometry, went on to become a faculty member, moved out west to the University of California at San Diego, where we spent the last decade with one singular problem, and that was how do we take a mass spectrometer and make it go faster? And the back of the envelope calculation was that we'd have to accelerate this by 2 orders of magnitude. The entire process would have to go about 100 times faster for this really to be sort of palatable across large discovery, meaning be able to run not hundreds or thousands, but hundreds of thousands of human samples to really uncover those markers in blood that provide maximal information. We spent the last decade developing technology, both hardware and software, and that's ultimately what gave rise to Sapient, that we spun out approximately 3 years ago to democratize access to these types of technologies and these type of data.
Yeah, I mean, given your background, right, and everything that you've experienced leading to this very point, what's interesting to me is that that you'd want to go down more of the path of how do we decipher the information that we're getting, versus I guess the direct patient interaction. And so can you maybe explain a little bit of what benefits do you feel like you're getting from having your background overall, but also are there things that maybe or have seen that also influences the way that you sort of guide Sapient in the development of your product?
Yeah, absolutely. As I mentioned, I spent years with the privilege of taking care of patients and helping them. And unfortunately, I don't practice currently, but you're absolutely right. It's something I absolutely loved doing. It was an enormous privilege to be able to help others and to learn and to work with them. And no matter who you are on the planet, human health is one of those things that's fundamental to the human condition. And so being able to participate in that when people were at their most vulnerable was really something I enjoyed tremendously. In the end, it was really this frustration around the idea that while medicine is accelerating and changing, it's not changing fast enough. The last major leap, you could argue, happened 30, 40 years ago, 50 years ago with the introduction of the first antibiotics. And since then, it's been sort of an incremental creep. Now, when you think about other areas of technology, whether it be information processing, whether it be aerospace travel, whether it be astronomy even, these have had major, major advances, where there's stepwise changes that have occurred in these fields over the last 30, 40 years. And one could argue that despite all the money that goes into it, medicine hasn't changed in that way. Our ability to really understand human health, to be able to predict who's going to develop what diseases, to best and most efficiently treat patients, while it continues to get better, it's not getting better fast enough. And being able to sort of work in that space was really what excited me. And now I've never lost my interest for taking care of patients. And oftentimes, whenever we're working in particular areas or particular disease states, we find patients to speak to, because there's nothing quite as inspiring as talking to someone who's had a loved one who's had a particular disease. And unfortunately, there's way too many of us, both within Sapient as well as in our world, whose lives have been altered immeasurably by disease. And being able to work in that space again is a privilege, and we leverage the feelings of others to make us go faster and push further in this space.
Yeah, and I would have to also envision that, as you look at your 20 years of experience, what are some of the pivotal moments in projects that you have significantly influenced both your career path as well as, founding Sapient?
Yeah, absolutely. There've been so many of them, it's hard to recount, but what I can tell you, there's a couple critical steps that have occurred. As I said, one of our real technical challenges, how do you make a mass spectrometer go 100 times faster? So the first time we were able to do that and it didn't explode was kind of a special moment. As we ran the first 5,000, then 10,000, then 50,000 human biosamples in our instruments, and we started looking at the data, it was unclear how much of human disease, once again, we were going to be able to explain. And it became pretty apparent very quickly that it was even higher than we initially anticipated. And we were learning things about people. We were able to sort of identify individuals who are at risk for virtually every human disease by markers that were in their blood. And the corollary that I always explain to people when we're sitting around Thanksgiving dinner is that when you go to the doctor's office every year, they draw these two tubes of blood, these two purple tops of blood, and the doctor measures 20 things in those. We call it the Chem 20. And it tells us everything about your heart and your cholesterol and your liver and your kidneys. And essentially, the majority of human diagnosis of disease is based upon those 20 markers. Now,
Mm.
there's 20 to 40,000 things circulating in your blood. And so we're capturing less than 1% of the total information. And arguably, it's not the most important aspect of the information. It's just markers that have been around for a long time. And so the whole idea, once again, is, well, if we could, instead of measuring 20 things, measure 20,000 things from a human blood sample, there's a lot more we could learn, once again, about your health state, how your organs work, the world in which you live, and how we maximize your health as well as your sort of ability to treat you in a setting of a disease.
Yeah, and I would imagine at some point, and maybe it's even now, you have to be impacted by the level of compute power as well that's available. I mean, having worked at one of the world's largest sequencing centers in the United States myself. I mean, I remember, several times we would run into these sort of bottlenecks on the data, processing side and the ability to use the bioinformatics pipeline to get the answers that we want. And the great thing was, we could also pay for more and get more. But it was one of those things where you're constantly sort of saying, I need all this information. And then the limiting factor, on the flip side, always winds up seemingly to be compute power as well.
Yeah, absolutely correct, Don. So there's a number of advances that have occurred external to us, which are very much central to our processes, whether it be in technology around mass spectrometry, which you could argue has changed night and day over the last 5 to 8 years,
Mm.
completely sort of evolved in step gradient functions. And as you said, cloud-based distributed computing, computational power, the ability to handle very, very large data sets and make sense of that data in a meaningful way. These are external to us and obviously have impacted virtually every single aspect of medicine. And certainly we're at the receiving end of that. Without these advances, it would be virtually impossible for us to do what we do.
Yeah. So as you look at the overall, challenges and opportunities that you've faced, in leading a biotech company, what are some of the, key challenges that you have faced, in the company itself?
Yeah, it's a great question, John. When you think about it, medicine, even though we think of it as being an innovative area, is still a large battleship. And evolution in medicine is a slow process. Now, rightfully so. You can imagine there's regulatory frameworks around this. How we sort of approve drugs hasn't fundamentally changed. How we test drugs hasn't fundamentally changed. How we develop them and test them hasn't fundamentally changed. And that's probably for a good reason. At the same time, there's this need to go faster. And we saw this during the episode of COVID, right? Where we had to, as a community, develop and distribute vaccines that were developed much faster than virtually any other medication ever has been. And so you're absolutely right that there are key challenges. One of the biggest, I would say, is a cultural one. And that is this shifting idea in medicine that we can do it faster. We can develop drugs cheaper. We can deploy them in a more efficient way by understanding the host, by understanding the patient to whom we're giving the drug. This is an area that we call personalized therapy or personalized medicine. While it's sort of an idea that's been around a while, it's still very much a new idea.
Mm,
And it's something that there's a pretty large gap between concept and clinical implementation. We still tend to treat every individual with breast cancer the same way, or everyone with heart disease in the same way, when we know that there's huge variations from person to person and disease state to disease state. So evolving past that activation energy, being able to
mm.
cross over that hump, and really impose personalized therapeutics is really the next big frontier, and being able to do that culturally for both physicians, for healthcare networks, for providers, for pharmaceutical companies is a big cultural divide that we have to get over as a community.
Yeah, I would imagine, as well, just that change in mental state. There are a few of you, I feel like that I've talked to on this podcast where you're really trying to change not just the thinking around the device or the technology that you're trying to develop. You're actually trying to shift the market, which is an entirely different problematic challenge that you have to face as well. That oftentimes, like you said, I mean, hey, if the regulatory bodies around us and everybody else that we're working with, is moving at a different pace, you realistically have to prove the much better state, I would say, of, where your technology is about to take people. And then not only that, but, just also the advances for patients. I mean, my goodness, if there was one big advantage to come out of this, it would be, hey, look, something's afflicting me. The 20 biomarkers aren't necessarily telling, all the full story. Let's look at the broader picture at what actually might. But it does lead me to my next question, which is so at Sapient Bio, for anybody that doesn't know, could you just explain a little bit about what Sapient Bio does and how you're really pioneering inside of the field of drug discovery?
Absolutely, Don. So I appreciate that. So Sapient Bio is what we call a CRO. We're a contract research organization. What that means is that individuals, whether it be large pharmaceutical companies, early biotech, foundations, and nonprofit academics, government organizations, they come to us to help them do the hard work of drug development and discovery. Now, what we do is we provide mass spectrometry resources in that we are able to analyze samples, whether they be blood samples, urine, tears, CSF, tumors, cells in a dish media, animal samples, whatever the case may be. We have the ability to generate large amounts of data. In these biological samples very, very quickly. Now, what Sapient does is we do two things. We generate that data, and then we also provide deep statistical analysis of the data. The last thing we want to do is return to someone a hard drive of zeros and ones and say, good luck with it. That's not our mission. Our goal is actionable knowledge. We want to be able to answer biological questions for our clients. And so in doing so requires us to generate massive amounts of data, both metabolomics, lipidomics, and proteomics in biological specimens, and then be able to provide statistical analysis of this underlying data in a way, once again, that answers questions for our clients. Now, you asked a really important question about how we actually shift this mentality, and we're at a very special time right now. Because we're in that post-COVID era, so many things have changed, whether it be subtle shifts or much more dramatic shifts in thinking, and that people finally believe that there is a better way.
Mhm.
Regulatory systems, whether it be from the government, are pushing to develop drugs faster and cheaper and make them more efficient. Pharmaceutical companies are very interested in being able to be more efficacious in their therapeutics. And most importantly, patients and patient advocates are finally pushing for a better way. And when you put all of that together, it becomes a perfect storm, if you will, for change. And I wholly anticipate that there'll be a remarkable amount of change going forward here in the next several years.
Yeah, it was one. I've made this comment several times on the podcast. I actually was telling somebody recently as well that I was corrected a little bit on this. That uh, I honestly, given the rapid deployment of the COVID vaccine as well as even just COVID test. I mean, we started out not even being able to detect the fact that somebody had COVID originally. And I remember one particular client that we were working with was saying, I want 100% of the samples that we're using in this particular study before there was a diagnostic. None of them can contain the COVID variant, and we were like. We have no idea how to find it right now. I mean, if you could find somebody that can, good luck. And so it was an interesting time, but watching the rapid approval, I just really thought, hey, look, we're going to walk away from here having a different approval process for drugs or a different sort of pathway. And I feel like, at least as an outsider, again, watching a lot of people going for drug approvals and things like that, that it is pretty much the same. However, I've recently been told that there are some significant improvements that people have seen on the clinical trial side as well, which is great to hear.
Yeah, no, absolutely. Things continue to continue to improve. It's a slow moving battleship.
Yeah.
Even through COVID, the empowerment that patients felt to go to the pharmacy and buy a diagnostic test and then take a swab and diagnose themselves with COVID. Since pregnancy testing, that's not something that's been around. That's just not something we've done. You go to your doctor to get a diagnosis. And the idea that my mother can go to her local pharmacy and diagnose herself is really quite a shift in the underlying thinking around medicine. And I think that empowerment of patients is only going to continue to accelerate forward change in medicine.
Yeah. Well, thank you for that. My next question is, could you share an example of any sort of recent breakthroughs or projects that you've had at Sapient Bio that you're particularly proud of?
Oh, absolutely. Again, we have the good fortune of working with dozens of clients across pharma, biotech, foundation, and nonprofits, and each one of these have success stories. Now, these are not our success stories; they're the success stories of our clients. But I can share conceptually a number of these. We have the good pleasure of working with a number of nonprofits, for instance, to understand maternal health in the developing world using very large studies, large epidemiologic studies across tens of thousands of mothers in which blood samples have been obtained. And we can try to diagnose at the earliest stage, or their markers in blood that tell us about the health of the mother through their pregnancy, as well as the health of the child. And that's one huge area in which we work in. We do quite a bit of work with large pharmaceutical companies and helping them to understand their drug development pipelines, how we can accelerate both the development of the drugs as well as the deployment. And there's a number of examples whereby we have the pleasure of working with large pharma companies for a drug that's failed in a clinical trial. And
Hmm.
we've been able to diagnose and develop a marker that tells us that subpopulation within the larger clinical trial that did actually benefit from the drug. And go back and in doing so reinvigorate a drug that presumably had been failed and would have ended up on the drawing board floor. And so those are sort of a number of conceptual sort of areas. There's many other diseases for which we now are developing early diagnostic tests for, whether it be diabetes, rare infectious diseases, et cetera, et cetera. And you hear about this now in the literature with GRAIL and cancer. But this is something that's going to become much more prevalent way beyond just oncology, but to other therapeutic areas for heart disease, Alzheimer's and neurodegenerative diseases, liver disease, lung disease, GI illness, et cetera, where we're going to push diagnosis earlier, and we're going to have diagnostic tests that tell us this is the best drug for this particular individual.
All perfect as well. The in terms of mass spectrometry, right? I mean, this is not new technology that's out there. I guess how does Sapient Bio differentiate itself from other mass spectrometry companies, and how do you foresee that things like computational biology will play a role into this in the future?
Yeah, absolutely. So there's a couple technical advances, once again, that have come in the field of mass spectrometry. And mass spectrometers have continued to get better and better year over year in a way that may not be fully appreciated, but arguably the technology has evolved as quickly as next-gen sequencing has in parallel sequencing processes. And so if you look at mass spectrometers today relative to 10 years ago, there's night and day difference. It's just not the same world anymore. And what mass spectrometers can do today, their reliability, their resolution, their ability to actually monitor and measure the dynamic range, all these technical parameters around these instruments, they've really improved massively. Now, our organization is particularly good at leveraging and using these technologies. They still tend to be somewhat of a dark art, particularly in making them go quickly and making them go fast, which requires not only fast mass spectrometry, but what we call chromatography, which is an area that we've innovated quite substantially in. Now, to your point, Don, to give you an example now, once again, from a blood sample, instead of measuring 20 things, we're measuring on the order of 20,000 things. And this includes 15,000 what we call metabolites and lipids, as well as 5 to 10,000 what we call proteins in that biological specimen. And being able to take that massive amount of data and say, among these 20,000 things, these are the one or two that are the most critical for disease X or Y, or for drug A or B, is really a computational exercise. And this is where having distributed computing, as you mentioned, becomes really quite critical. Being able to do this across very large populations, which is what's required in order for robust discovery, as well as robust validation of discoveries, is really central to this. And to your point, once again, this is why we have as many chemists as we do computational biologists at Sapient. It's a lesson we learned very early on in our evolution, that we'd have to be able to provide the statistical analysis of the data as much as we'd have to provide the ability to generate that data.
So knowing that I can't hold you to any of this, but I would love to hear your view of the future, right?
Yeah,
If you had a crystal ball and we
yeah.
fast forward, let's say 20 years into the future, and we're looking at Sapient Bio now and the impact that it's having on patients and things like that, what would you want to have happen in the future and based on the technology that you guys are developing?
Oh, it's a really great question. And here's where I'm going to get into trouble, because I am a blinded optimist, if you will, and that I see the potential of the universe and the world, and really what can come out of this. And I hope this evolution is realized. But I can imagine from a patient perspective with regards to diagnosis. Diagnosis is going to move years in advance. It no longer will be that you have diabetes or heart disease or Alzheimer's, but 20 years prior to that, you have a high risk of diabetes and heart disease and Alzheimer's. And that will allow us to institute therapy at the absolute earliest time point, when we know we can really effectuate disease processes, as opposed to after disease has started, when it's much harder to treat. It's much
Mhm.
easier to prevent the car from breaking at the earliest stages, as opposed to once the car is broken, fixing it. And that's the way the human body works as well. And so that's one major area that I suspect is going to shift. I expect that's going to shift more so from a physician's office to patient derived or patient centered diagnosis, where because I would like my blood tested, I can go to a laboratory and have it tested, as opposed to needing to visit a physician and there being a layer in between here in my actual personal health care. From a drug development perspective, you've seen the startling numbers, and you're well aware of them. It costs you, north of $2 billion in approximately 10 years to bring a drug to market. And that's an absurd cost, frankly. And it's not because anyone likes to waste time and money, but it's an extremely expensive process that's fraught with failure. It's
Mhm.
one of the few fields in the world in which you have a 90% failure rate. And if you can hit 10% success, you're doing great. And not even in baseball can you get up there and swing, 10 out of 100 times and call it a success. And so being able to take those numbers and completely shift them. We need to be able to move clinical trials in a way in which we're no longer treating broad diagnoses such as diabetes, that we know represents 5 to 6 different disease states, but rather a subset of disease in which the efficacy in that subset is far higher. This is going to improve overall patient outcomes. This is going to make drug trials go faster and be much more efficacious. And I really hope we get to the point where drug development is no longer a 2 billion 10-year process, but is somewhere in the order of a 200 million 3-year process. And I
Yeah.
don't think that's absurd. And yeah, that's sort of where I hope this field evolves. And hopefully 20 years is not where we have to wait, but rather in the next 5 to 10.
Wouldn't that be nice, right? I mean, it'd be great to be able to fast forward, to that point as well. So, a few questions that I'd really like to hone in on to focus on you. What's the greatest leadership advice that you've ever received?
Oh my goodness. Wow. That is a really good question. My leadership philosophy has always been my job is to surround myself with people who are far smarter than I am. And then I view myself as the glue or the grease. It's my job to bring people together. It's my job to provide grease to make the wheel go faster. And if you bring really talented people together in a room, who are all excited to work together on problems larger than themselves. And you put a little bit of food in the room and you get out of their way. Usually they'll end up solving it in a way that certainly is beyond my ability. And I think that's the sort of foundation of Sapient. I think everyone is equal, but everyone is not identical. And
Mm.
much of leadership comes down to understanding how you best support each individual person on a team. And we all have slightly different needs. And if we can do that properly, um, if we all have a central vision and we all row in that same direction, that's when amazing things happen once again.
Yeah, absolutely. What inspires you?
I think the opportunity, sort of what is possible. I can imagine what it was like to be alive in the 60s and 70s and talk about going to the moon, right? The moonshots, the things that, fundamentally humanity is different the day after that occurred. And we have not had a moonshot in medicine. Now, I know we had the cancer moonshot program and we've other moonshots, but we have not had a major, major victory where fundamentally our ability to diagnose disease and or treat it has been altered in the last half a century. Since once again antibiotics, intensive care unit optimization, these sorts of things, logistical operations, but on the development side of drugs, that hasn't changed in quite some time. And so the opportunity is massive here to really again impact a fundamental aspect of humanity, and that's our health and our ability to live and be in the world in which we would like to be.
Yeah, thank you for sharing that. What concerns you?
The path is long and it's fraught with challenge. So much of what not only we do, but life sciences is very much subject to macroeconomic forces and headwinds and tailwinds and the world, again, in which we live. Um, and so there's a practical aspect to this. There's a pretty big gap between inspiration, having technology, and having a successful business, and being able to sort of live in the stars with your feet firmly planted on the ground is really, really important when you're in this space. And we're seeing a massive shift in the biotech sectors now as funding and capital is drying up and sort of the metrics are changing pretty substantially relative to the last several years. And so just being able to ride that wave and continue to being successful is a challenge that we have to be very cognizant of.
Absolutely. What excites you?
Oh, I'm a simple guy, Don. I get to go to work every day and play with amazing toys and hang out with people who are far smarter than me. Like,
Mm.
that's a fun job, right? And again, solve really, really, really hard problems with really smart people. And if I always say, whenever we're recruiting folks to Sapient, if that idea doesn't excite you, this is the wrong place to work. And I'm very honest about that. But for me personally, that idea is what gets me up every morning. And I don't think that'll ever get old.
Yeah, absolutely. Well, Dr. Mohit Jain, thank you so much for coming on the Life Science Success Podcast and for telling us about Sapient Bio and all the great work that you're doing there. I'm very encouraged to hear all of the things that you're doing around mass spectrometry and with this technology because as great of a platform as it has been, it definitely is needed an update as well, at least based on the experience that I've had and. I look forward to seeing, all the great things that you guys are working on.
Thank you so much, Don. For any of your listeners, please feel free to reach out to us. Our website is www.sapient.bio. We are always excited to chat with folks in the pharmaceutical and the nonprofit world, and there's so much that we can all do together.
Thanks a lot.